It's great to be back at the JGI. I was here for 3 short years, 2002 to 2005. I was at Berkeley, and I want to thank Eddie for generously supporting me. Some of the ideas that you'll hear about today are actually incantations of the ideas that I had back then. Basically, I completely left DNA sequencing because I'm really, as a scientist, I'm interested in beautiful de novo sequences of genomes that we've never seen. I mean, as a informatiker, that's kind of my goal. And so I just, with the short read sequencers, I felt we kind of lost it. It was beautiful for medicine. I mean, and I understand why it happened, but it kind of, as a technology developer, I just didn't feel that the mathematics was there. So in 2012, when I went to Germany, I had a little bit of spare coin in my pocket, and I was thinking about what to do with it. And I ended up talking with Mike Hunkapiller about where PacBio was going with the long read sequencers. And Mike said a couple of very important things. So actually, this is the outline of my talk. I'll talk about, is perfect assembly possible? I'll talk a little bit about the current state of affairs, and then I'll talk about the things that I think are the most important in trying to get to perfect assembly, which is dealing with the artifacts and the reads, and with dealing with repeats. I probably won't get to dealing with repeats, but we'll see. If I get there, I do. But what he said in the con call that we had that really got me excited is he said, so I knew the reads were long, and I knew they were noisy, okay? But he said he said random in the two magic contexts, which is that the sequencing errors were basically random, and that the read sampling of the underlying genome was effectively random. And as a mathematician, I knew immediately what this meant, and I got incredibly excited, okay, because what it meant to me was that basically perfect assembly is possible, okay, in principle. These are the characteristics that we need, and in fact, we've never actually had a machine which has these characteristics. Okay, and they're not perfectly random, but they're close enough that from a mathematical perspective, the early theorems that were actually worked out in the 80s actually work out. And in fact, I got so enthusiastic that I'd been shamed into getting into tweeting. And so I tweeted, one of my first tweets was this 140 character theorem about why I thought perfect assembly was possible, and I'm going to explain that theorem to you now. So basically, what does it mean that the random sampling is Poisson, that you're basically grabbing out? What it means by actually a very old theory from Lander and Waterman from 1988 is that as you sequence more and more, you will cover more and more of the genome probabilistically until at some point you've got the whole thing covered. And in fact, you can pick some level of coverage, 20, and if you do enough, you'll eventually make sure that the entire genome is covered 20 times, right? And I'll kind of leave off for how much is enough. But it means basically, if you're willing to work hard enough, right, you can have any level of coverage across the entire genome that you would like, okay? So say you want 10, okay? The other thing is, what does it mean for the read error to be random? What it means for the read error to be random is it means that the Q-score, which is the log of the likelihood that it's wrong, of the consensus of K sequences increases linear in K. This is an old theorem, again by Waterman, and his then postdoc at the time, Churchill, from 1989. And basically, what it means is that if I do enough sequencing, I can guarantee any desired accuracy. In other words, forget about Q40, I can have Q80 consensus if I want, right? So I think this is quite clear, right? That basically, if I'm willing to pay enough, or sequence enough, or wait long enough, right, I can basically derive a complete covering of a genome and produce a consensus at any level of accuracy that I want, okay, with this technology. Of course, I'm ignoring the most important thing, which is what about those pesky repeats, right? Because, I mean, that's really the problem, right? It's the fact that if the genome were a random string, it'd be easy, okay, we'd be done. I could leave now. But it's got those repeats. So basically, what in effect I did at Celera with the Celera assemblers, I developed what it called the string graph idea. So here's a little small genome. So imagine the yellow line is some genomic sequence. And imagine that each of those colored arrows represents a repetitive element. Okay, so there are 2 copies of the purple element, 2 copies of the green element, or 3 copies of the green element, and 2 copies of the orange element. Okay? And imagine that those copies are actually identical. Okay, let's keep it real simple. They're identical, and they're longer than your read length. Okay, so there's no hope that you can get through them. Okay, now basically, I'm not going to bore you with the string graph, but basically, imagine annealing all the orange repeats together, and I'm annealing all the green ones together, and annealing all the purple ones together. And imagine that the yellow string in between, the unique stuff in between, is a string. So it's floppy, and you can flex it. And what do you get? Okay, you get this. Okay, that's a string graph. Okay, that's it. You got it. Okay, hopefully that was simple enough for everybody. Okay, but what's really important about this string graph, and what we realized kind of at Celera is that most of the time, the yellow stretches are pretty long. And if you have a long enough yellow stretch with enough reads covering it, by simply looking at the arrival rate of the reads, you can tell definitively that's a 1x part. That's a unique part of the genome. So I can identify all those yellow bits as being 1x, okay? They're covered once, okay? So I can identify all the yellow bits. And if I can identify the yellow bits, then I even know how many times I go through all of the copies, okay? Because of the number of ways in and the number of ways out, right? The green one, there's 3 ways in, 3 ways out. There's got to be 3 copies of the green guy, okay? And then I know that I have a beginning and an end. In this particular case, it's not circular, but it also works for circular DNA. But basically, then the answer, and this, in effect, is the answer to assembly. If all I had was reads, okay, no mate pairs, no nothing else, if I just had the reads, and these repeats were longer than the reads, then this is actually informatically the very best that I could hope to do, okay? And it is the answer to assembly, okay? But what do you want? You want a linear string, right? And so, okay, you can say, well, how many Eulerian tours are there, right? How many ways can I walk from the beginning, from this guy here to this guy here? Are you seeing the cursor? Yes? Oh, it doesn't show the cursor, crap, okay. All right, well, you see the 2 ends, right? And the idea is how many tours are there? Well, it turns out there's actually only 2 tours. Okay, so with one appropriately placed PCR experiment, I could determine which of the 2 tours was right. Of course, or I could add mate pairs or something else, okay? So in a sense, that's it. But what's the reality, okay? So I said assume that those repeats were perfectly identical, okay? They were identical. And in fact, it'd be a lot easier, the problem would be a lot easier if the repeats actually were identical, God damn it. Okay, they're not, okay? They're actually, they have microheterogeneities within them, right? There are some that are prefixes and they've been truncated, and there are all kinds of things that are going on, right, in the evolution of any repetitive element, okay, unfortunately. So actually the reality is, the ideal that I would love to see is I have a bunch of ways in, I have the repetitive element that's been over-collapsed, and then I have a bunch of ways out, okay? Well, the reality is I have a repeat hairball there, okay, where all of the small differences are making basically a kind of a collection of paths that go from one end to the other. So in some sense, the problem is I got to get through this repeat junction box, right? I got a bunch of ways in, I got a bunch of ways out, and I know there's this junction box. And the question is, how do I get through there? Okay, so in a nutshell, that's kind of a way to frame the repeat problem. That's what you got to do, okay? You guys with me so far? Yeah, is it pretty good? Okay, I got enough nods, I think I'll proceed. Okay, so, well, one of the nice things about the repeats, the long reads actually, is that a lot of the times you'll see repeats being spanned. So suppose that blue bar there is a repetitive element, okay, one of the nasties, and the orange thing is a PacBio read. It's a long read, okay? So that could be like a full-length line element, an 8,000. The blue thing could be an 8,000 base pair line element, and the orange bar is a 12,000 PacBio read, okay? Well, so suppose you want to try to get through this junction, right? So now I've kind of coded, so the orange guy is supposed to go with the orange guy, so you know what the solution is. Well, if I've got a read that spans, the repeat, right, and it has enough unique flank on both sides, then I know that that read goes there, right? And in fact, I know that the orange way in goes with the orange way out, right? So it solves that particular path through the hairball, okay? And in fact, if in general your reads are long, okay, then your expected read length minus twice whatever your kind of minimum flanking length. So for this technology, about 1,000 base pairs is more than enough. Should suffice. So with enough reads, okay, and a flanking length of 1,000, and say I ask the R25, I should be able to resolve, R25 under our hands is about 22 kb. I should be able to resolve most repeats that are 20 kb or less. Okay, so that's one of the nice things that you already know that about PacBio, long read, or MinION. Okay, by the way, I'm agnostic. I'm perfectly happy to use nanopore when it's at a scale that we can do big genomes, and when the error is under control. So basically, you know already that long reads mean that a lot of the repeats go away, and this is why, right? Because basically they're spanned. And spanning means I know how to get through the junction box, okay? But what about if I had a junction box that was so long, okay, that there's no hope? Now, it's 50,000 base pairs long, okay? And we see these, by the way, okay? As you know, there's a geometric distribution of repeat lengths, right? And so there are big ones in every genome. Okay, and so is there a way to get through these things? Well, now I'm going to be happy that the copies vary, because if the various copies vary, they have microheterogeneities kind of along them, okay, then I can basically use those heterogeneities to identify unique signatures across it and actually finding a tiling path that's unique to one of those particular paths and separate out that pair, okay? So it's basically like haplotype separation, except I have to do it K ways instead of 2 ways, okay? And it's basically exactly the same idea. And by a work I did way back, I presented in some obscure Japanese conference in 2002. I basically showed that if you have a microhet if you have 5 differences per read, even in the presence of 15% error rate, you can still walk your way through. So for a PacBio read that's 20 kb long, that means all I need is to have a microhet in the repeat every 3 to 4 kb, and I should, in principle, be able to get through it, okay? Now, this is all a theoretical talk, because I haven't done any of this stuff, okay? I've only done a little bit of it, okay? But I wanted to basically convey, like, why, like, the light went on and why, I'm back in the game again, okay? Why am I back here, okay? Because I'm doing microscopy and computer vision, right, for developmental biology these days. And in fact, I personally now have gotten back in because I got really excited about this. And now, the point of the matter is I've never said here how much do you have to do, right? And so obviously the game is to build an assembler that's efficient in the sense that it doesn't require too much coverage, okay? And that doesn't require too much computer time, and it deals with that 15% error rate. So in other words, the error rates, because see, a lot in the early days, I think this crowd is pretty sophisticated now, but in the early days everybody said 15% error, that's crap. I don't want to use that instrument, okay? And that was completely wrong, okay? That was completely wrong, okay? The error rate is only a function of efficiency, right? Can I write computer programs that operate on such data, right? Because all the assemblers are assuming a 0.5% Illumina error rate these days, okay? Which means that we have to go back into the trenches, right, and rebuild all these things, which is kind of fun. So what's the current state of affairs? Okay, so an assembler is basically a pipeline of modules, right? So this is kind of the generic long-read assembler. You basically try to figure out who can overlap with who. Then Jason introduced this idea of you try to correct to get rid of that 15% error rate, to get it down to 1% or something reasonable. Then you overlap again, except now at a higher stringency, and then you can even use something like the Celera assembler and then run a consensus algorithm. So the first one that you saw a few years back was HGAP, which basically used a tool called Blazor for the overlap. Jason wrote the corrector. The Celera assembler is, some old thing legacy thing that still seems to persist today somehow. And then there was Quiver that was written by David Alexander at PacBio. And the most recent tool that most people are using are Falcon, and this is where kind of you'll see finally there's a contribution from this guy Gene Myers, which is the Deliner tool, which basically made it about 25 times faster than Blazor and makes it so that you can do like a human genome in 20,000 CPU hours instead of 400,000 CPU hours, which is a big gain. So, but the point of the matter is that I'm basically doing this work myself, so and it takes time to build good modules. An assembler is not a simple thing. So basically, strategically, a big mea culpa. I'd love to give you an assembler today, and I'd love to be able to take every idea in my head and put it into code and, and give it to you, but it's a painstaking process, okay? And so my strategy basically is to release useful modules as I develop them. So this is kind of the infomercial part of the talk, okay? If you are interested in what I'm doing, just remember Dazzler blog, okay? And Dazzler stands for the Dresden Assembler, okay? All right? And I mean, I am in Germany now, right? So it's the Assembler. And okay, and I'm using a blog in a very weird way. There's no opinion in here. There's just digressions about assembly, and I try to present the ideas with examples. And it's basically a vehicle that I'm using to distribute my open source software. Okay, and one of the important things is, and so the blogs are basically kind of about the things that I'm building and why I'm building them and how to use them. There's a command guide which has actually got a reference of the things that are actually out there. You can go to those command guides and you actually get, for example, Unix command line descriptions of everything that you can use. And most importantly, there's a link that says where to go to GitHub to actually get the code, okay? And if you're willing to follow me on Twitter, I tweet like once every 3 months, okay? Usually when I have a new blog post, okay, introducing a new thing, I tweet the updates to the blog, okay? That's mainly what I use Twitter for. Okay, so let me just finish by saying that, I think you guys already all know this, but current assembly is already competitive, okay? So for example, melanogaster with the old, P4C3 chemistry, they sequenced it to 100x, and they basically got a result that's very close to release 6, which is, and Drosophila is by far the best finished genome we have, right, in the business, okay? So this is pretty amazing, right? I mean, you just out of the box with Falcon, which is a very good assembler. I work with Jason a lot, but it still doesn't achieve my concept of perfection. We're getting results that are already, okay, competitive with the best finished genomes, okay? For human, it's, okay, so now we get up to a bigger scale. Well, not quite as good, okay? You can see the PacBio 84X has quite a few more contigs, and the stats aren't quite bad, but it's missing some of the genome, about 180 megabase pairs, but it's still pretty damn good, okay? And in fact, if you do something like BioNano, the BioNano maps aren't any better than the PacBio maps, but when you combine the two, okay, you get pretty damn close again. Okay, so with a scaffolding engine, okay, and the PacBio, and with current technology, you can produce, in principle, okay, publication-ready genomes that stand the test of time, right? I mean, wouldn't it be wonderful if we had every genome in the world perfectly, so it never had to be done again, okay, in GenBank, okay? I mean, it might be possible, right? Okay. I'll be gone, but we'll get there. And for example, as an example of my own work, we're working on Schmidti mediterranea, a regenerative organism. It's 70% AT rich. It's 800 megabase pairs long. It's exceedingly repetitive with lots of microsatellites, and we have a 60x data set. And with that data set, we've been able to get an N50 contig size of 750 KB, almost a meg, and compare that to the 10 KB that was achieved with Illumina, okay? All right, I don't think there's much difference, and that's with the Dazzler, and in fact, which is, I have an operating assembler, but I'm not willing to give all the parts to you simply to protect you, okay, because you'd die if I gave you all the parts. There are a lot of caveats, a lot of stuff still cached. The stuff that I put out in the public domain, I beat on it hard, and I'm only gonna give you the things that I think are really ready for public consumption. But the intention is to give everything, okay? And then we've gotten very interested in using Hi-C for doing the scaffolding. Works great, okay? So we now have Schmidti into 300 scaffolds with a 4 megabase pair N50, and we're hoping to publish it very, very shortly with a very interesting story about the repeats, by the way. Okay, so let me then, how am I doing, Susanna, good? 15 minutes? Okay, 10 at least. I'll at least be able to do artifacts, okay? Because I think this is an important story, okay? So the story goes back actually to here when I was here in Berkeley. So I actually wrote kind of the seminal paper on string graphs in 2005. And part of that paper, what I did is I took C. jejuni and I I created synthetic 700 base pair Sanger reads with no mate pairs. Remember, this is quite a ways back. And I produced 10x, and I produced the string graph of it, and I got that beautiful result there, just like the picture that I gave you, right? It was pretty cool. I was very excited, okay? Then I took and the assumptions were that the reads are contiguous, and then I took the real data and I ran it, okay? And I got a hairball, okay? It did not look like that at all. It was a mess, okay? So why was it a mess? Well, it meant that something was different between the real data and the simulated data. So what was true about the simulated data? The simulated data was actually a piece of C. jejuni, right? A contiguous piece. There was no, okay? And I had introduced 5% errors into it. So it didn't have too much error, it didn't have too little, it had 5% error. So it had a uniform error rate, and it was a real piece of the underlying genome. What's the problem with real data? Chimeric reads, right? Unremoved adaptamer, contaminants, right? And excessively erroneous reads, okay? In other words, ones where the Q values weren't correct, okay? By the way, Q values are not a guarantee, okay? And so what this means, what I came away with from that is that, man, if I was ever really going to be good at this problem, I was really going to have to take care of those things. That taking care of those things was absolutely essential. There was nothing wrong with the string graph theory. The string graph is perfect. You give me a collection of reads that are a slightly perturbed copy of the stretches of the underlying genome, and the string graph gives you a beautiful result. The only reason you don't get that out of the data sets that you're putting in there is because they got that stuff in them, okay? So it says that the problem's been underappreciated. So you want to scrub, and what does a scrubber do? A scrubber basically is something that edits the reads so that at the end of the day they have exactly the properties that you want, that they represent a contiguous stretch of the underlying genome, and that every part of an edited read is of a reasonable quality, in the case of PacBio less than 15%. And if you do that, then you will get a string graph, okay? Okay? So, and in fact, it's interesting because there are papers about scrubbers, about tools that, try to figure out how erroneous the read is and fix it up and da-da-da. But nobody's ever actually built one that takes care of everything, right? And really takes it seriously, okay? They were all kind of like side papers, right? No, okay? If at one point I'm going to come away with today, this is the game, okay? Besides the repeats, this is the game. Okay, it's all about the quality of what you put in there, and I'm not jamming the people that do the sample prep. I mean, you're gonna, you do blunt end ligations, you're gonna get chimers. Okay, all right. At 15% error, you're gonna miss the adaptamers. Those things are gonna happen. Okay, but the point of the matter is that if you're really serious about building an assembler, you got to take care of them. Okay, and in fact, with PacBio, it's even worse because let's take a look. This is a read. This is a schematic of a read. The x-axis is supposed to cover about 25,000 base pairs, right? And what I'm plotting is the error profile, right? The error rate, okay? Look at the error rate on that thing. It runs about 10 to 15%. Sure, that's the average, but it goes up and down. There'll be 1,000 base pairs that's 8%, and then there'll be the next 1,000 will be 15%, okay? Then there'll be a stretch where for 20 base pairs it just goes, ape shit, okay, and spits out a bunch of A's, okay? And then a little bit later on, what'll happen is another piece of another Pol molecule in the well will start up a reaction, and for the next 3,000 base pairs, you're going to get a little musical jamboree, right, which is going to completely confuse everything because it's basically effectively random. But you don't see any change in the Q scores. There's no indication from the Q values that come out of the machine that that's going on. Okay, but it's complete junk. Okay, and then suddenly that guy shuts up. Okay, and it goes back to the original Pol molecule, and it's perfectly good. Okay, so you want to use the data. You got to figure out that that's going on. How many of you have a tool that will tell you that that's going on? How many of you have a tool that will tell you it's 15% for the next 1,000 base pairs and not 8%? Okay, need that, right? So I'm going to show you how. Okay, so. Sorry, I'm being maybe overly dramatic, but I get excited. Okay, anyway, so the profile is not computable from the instrument QVs, and it's nothing wrong. The guys are, Alexander and those guys, they're really good. They did exactly what Phil Green did, and the technology's right. But frankly, with a single molecule and the SNR that they're having, they don't have enough information to really give you a meaningful quality score. And it's completely different for each read. So what do you do? So basically, one of the nice things about the liner is the liner basically finds every significant local alignment, not an overlap, but a local alignment, which means any two kind of pieces, right, that have a significant statistical overlap, right? So basically anything over 1,000 base pairs, right, that matches at 30% difference between the two, it finds it, and if it's a piece of the read, that's fine, it finds it. This turns out to be very important. It's a local alignment, okay? And it finds all of them. So imagine then that you have a read, one of your reads, and you've compared it against every other read in your data set, okay? What I'm going to show you then is the intervals of the other reads that overlap, that align with that read, okay? So these are the other reads, okay? And the brown tips, basically, if you want to understand, so the white intervals are the intervals that aligned, okay? And the brown tips are the pieces of the B read, of the other read that didn't align. It basically says, look, I stopped aligning with you, but I keep going, okay? And I just draw a 500 brown tip, okay? Whereas at this end, right, it's white at the end, which means that was the end of the B read, right? And I'm at 5. Okay, great. I think I can at least make the point about artifacts. So the first thing is I found these, I call them pylograms, okay? So it's for each read I have a pylogram out of the liner. And I can look at these things, and that's kind of normal, okay? But look at this guy, okay? You can immediately tell that this guy's telling you need to trim the first so many base pairs, right? Nobody aligns with the first 500 base pairs of that guy. What about this guy? Oh, look at that. There's that big kind of divot there. What's going on there? Well, that could be the signature of a chimer, a missed adapter, or it could be an exceedingly low quality region where just there's no alignment because the data is too poor, okay? And then there's this, okay, which tells you immediately that must be a repeat, right? So it even gives you a signature of a repeat. So it's pretty interesting, right? Just by looking at how all the data aligns up with itself, okay, you get interesting information. Now the trade-off is that it's expensive, right? You have to do a huge all against comparison. But look, do you want it fast or do you want it right? Okay, in the case of assembly, okay, you want it right. I mean, who gives a dang how long it takes, as long as it's feasible, okay, all right? All right, what you want is right, because you just have to run the thing once, okay? And as long as it puts out the thing, that you're done, okay? You can go home. So I think in order to skip, I gotta kind of get everything. I've got a very interesting way. I was basically thinking about how to store alignments, how to store these huge alignments. And BAM is an atrocious format, right, for this. You would not want to. So I found this kind of short way called trace points. And the interesting thing about the trace points is that for every 100 base pairs of the A sequence, they told me how well the B sequence aligned with it. Okay, and you can keep that information very tightly, okay? 0.02 n bytes for each overlap. So these files get big, but not too big. And so what that means is if I come back to this pile, right, you don't see too much in this pile, okay? But what I'm going to do now is for every 100 base pairs of the A sequence, I'm going to show you how well the B sequence aligns to it, okay? And this is kind of for free in my technology. And I'm going to get a little heat map where the worse the alignment, the redder the score. Okay, I mean, the first time I saw that, I got pretty excited because I immediately understood that the beginning of this read must not be very good, right? Because, there's occasional red dots, but look at everybody here is basically red, right? What does that mean? If everybody that aligns with you doesn't align with you well, what does it say? It says, you're the rotten guy, okay? Okay, you are the rotten one, okay? And basically everybody says, you're no good, okay? And it's not so very high quality here. So I do a very simple thing. I basically just take the average over the lower quartile with respect to the coverage, and I basically call that the intrinsic quality value. Okay, so it's a statistic. It doesn't mean anything other than that it correlates beautifully with how good that part of the read is, okay, beautifully, all right? And in fact, I use a color scheme, a green, yellow, red scheme, where I look at the distribution of these values, and I say the top 80% is good, the bottom 5% is bad, anything in between I'm not sure, and then I color it, okay? And what's fantastic about this is now you can actually start looking at your data, okay? And I don't know if you guys probably have never seen this stuff, but you tend to think, oh, I've got this read and it's got a certain quality. Okay, I mean, look at this stuff. I mean, look at that guy. You don't want to use that guy at all. That's a failed read, okay? But you don't know that, right, unless you've done this exercise, okay? And that's the beauty of this. So basically, I built a scrubber out of this thing, and it's interesting. There's basically 1.4% of the reads have unremoved adapter. 6.5% of the reads have a chimeric gap. Okay, which if you didn't remove would be problematic. And then I did a certain amount of scrubbing. In fact, so here's the deal. Here's what kind of these piles look like zoomed out before scrubbing, and here's what they look like afterwards, okay? No brown tips, okay? Everybody aligns. In fact, but you do throw away quite a bit of the data, okay? About 15% of the data you get out of PacBio instruments should be thrown in the trash bin, okay? And there's no point keeping it. Throw it in the trash bin, it's fine, okay? It's part of the cost of doing business, okay? And so here's a scrubbing example. So as an experiment, what we did is we said, okay, so now that I know how to scrub, let's take a 30x PacBio data set. Forget about 100x, 50x. Let's take a 30x, kind of a cheap data set, and let's scrub it. So I throw away about 20% of it. I'm left with 21x, okay? I basically align that to K12, the K12 reference, and aligns beautifully. In fact, there's a little bit of color in here, but you can't see it. There's a little bit of yellow and orange if your eyes are good. So there's very little bad thing. And here's basically the alignment to everything, okay? And if you assemble without correction, okay, you get a circle, okay? So I think I'm going to just jump to the end, okay? I had a nice little thing about repeats, okay? But I'll save that for another day, okay? But I've got repeats on the run. I've got a way of going about 10% faster. Sorry, I don't know how to jump ahead. I'm just trying to get to my conclusion slides, Susanna, sorry. What I've reported then is that hopefully I've conveyed to you why I think perfect assembly is possible. I think probably all of you know that, but I wanted to give you a principled rationale. I introduced this idea of intrinsic quality values and the idea of scrubbing, and I think that I've tried to hopefully emphasize how important it is. And then forthcoming, we are going to build things that basically solve the repeat issue. And I think probably the most important thing is that you can get the liner components and all the things that support it, but very soon now, I'm ready to release actually the scrubber that I talked about and the repeat masker, which I didn't talk about, which will help you speed up your things. But in particular, I'm going to hopefully empower you guys so that you can see the quality of your PacBio reads, which I think is going to be an important thing. And so watch out for my next, quarterly tweet, okay? All right, and I think the last thing that goes without saying, frankly, is that while you guys are probably thinking about what you can do today, I mean, you should keep in mind that the cost of this technology is going down and down and down. And you don't have to be a futurist to see the day, probably a decade forward, okay, where for less than $1,000 you'll be able to de novo sequence a human genome with a long read instrument, okay? So think about what you're going to do with that, okay, because it's coming. Okay, thanks a lot. Sorry I went long, sorry.
That's all right. I think we can still tweet it when I post it. I'll ask a quick one because I can't resist. And my area is, microbial communities. And I was wondering, do your assemblers explicitly require uniform coverage? And what do you think the prospects would be for perfect assembly of organisms out of mixed samples of different abundance?
So sorry, I'm not sure I understood the question. You're just saying if you're unlucky and you have a low coverage region, what does it do there?
No, I'm saying if you have many organisms and they're all at different abundances in a microbial community.
I'm a bit confused about what to do with metagenomics because, intrinsically, you're right. It requires enough sequencing depth, right? And so, I think, I saw actually a poster from the Dovetail guys with a high C. I really think that metagenomics is going to require some way to concentrate, okay, and explicitly abstract components out of the sample, right? That just shotgunning it, right, blindly, okay, has its limitations, and I'm sorry I haven't figured out a way around that.
Okay, great.